Hello guys, so today we are going to study about AIDS. The full form of AIDS is acquired immunodeficiency syndrome. By the name we can know syndrome means a lot of symptoms, means a group of symptoms. Symptoms caused by deficiency of immune system or a deficiency of immunity in the body of that patient. Because of the deficiency of immunity, there are lots of symptoms seen in the body of patient. So that this group of symptoms is known as AIDS. How is it caused? It is caused by retrovirus, or also we can say human immunodeficiency virus. By the name we can know this virus is responsible for making deficiency of immunity in the body. So the major thing we can see in the patient is the T cell mediated immunity is suppressed, and there are lots of opportunistic infection and neurologic disorder. So what is opportunistic infection? Opportunistic infection are those kind of infection where the microbes are just looking for the opportunity, the right opportunity to attack the patient. That means if the patient's immunity is very low, then the infection will easily, the microbes will easily attack that person, and the person have to suffer from infection. So those are opportunistic infection. So how the AIDS is transmitted from one person to another are through sexual contact, through parental inoculation, through transplacental transmission, that means through placenta, means from infected mother to the baby, and via blood and blood product, and via other body fluid. Blood and blood product means if you are having infected blood from the blood donor, then also you can suffer from AIDS. What is pathogenesis? First of all, the patient will suffer from HIV through any route. Then the infection is spreaded to lymphoid tissues. Then by the lymphoid tissue, it is spreaded to whole body. Then our body will fight through the immune response. Then we can see a lot of microbial infection in our body because of low immunity system in our body, and also we can see a lot of viral replication and CD4 plus cell lysis. Then after a certain time we can see depletion in CD4 cells. There is a very less CD4 cells in our body. Then it causes AIDS. So there is one question: how CD4 cells are less than normal range in AIDS? That means what happens is the HIV virus binds to CD4. It binds to CD4 and it will replicate within the cell. So basically it destroys the CD4 cells, because of which there is less CD4 cells found in our body. So if you are suffering from AIDS, the lymph nodes of our body shows adenopathy, that means swelling of the glands, and there are lots of opportunistic infection. So what are the basic clinical features? We have already mentioned there are lots of opportunistic infection from bacterial, virus, fungal, protozoal, anything, and secondary neoplasm is seen like Kaposi sarcoma, primary CNS lymphoma, Hodgkin's lymphoma. These are just example. And lots of neurologic diseases are seen. Wasting syndrome is seen. Wasting syndrome means 10% of the body weight is lost. And persistent generalized lymphadenopathy means the patient will suffer from a lot of extralingual site lymphadenopathy of more than 1 centimeter for more than 3 months, and a lot of GI lesion and manifestation, pulmonary lesion, mucocutaneous lesion, hematologic lesion, CNS, gynecology, renal, hepatobiliary, cardiovascular, ophthalmic lesion, musculoskeletal lesion, endoplasmic lesion. So basically we can understand all of our body, the whole body will get infected. So all of the disease which can occur, which is possible to occur in our body, can happen during AIDS. So, for diagnosis, how can we diagnose if the patient is suffering from AIDS or not? First of all, we have to understand there is no any specific diagnosis. We can only diagnose after we get to know that the patient is suffering from a lot of infection or tumor or something like that. So, basically, we have for diagnosis, we can remember these three things. We can directly check if the person is suffering from HIV. We can check for human immunodeficiency virus. Then after, we can check if the immunity of the patient is low or not, because we know for the patient in AIDS the immunity is very very low. Then after, another thing we have to check is opportunistic infection. If the patient is suffering from a lot of opportunistic infection, it can also indicate the sign of having AIDS positive. So, for first of all, I said infection, HIV infection. For test for establishing HIV infection are, first of all, is antibody test and direct detection of HIV. Antibody test is ELISA and Western blot test. Direct detection of HIV are P24 antigen capture assay and HIV RNA capture method. So these things you should remember, I should say. So, next thing is DNA PCR, culture of HIV from blood monocyte and CD4 plus T cells. These are the tests from where we can know if the patient is suffering from HIV or not. Next thing I said was immunity. We have to test the immunity of the patient. So we have to test CD4 plus T cell counts, and we can see there is increase in CD8 plus T cell counts, and we can see decrease in number of cells, and also number of immunoglobulin is increased. Why the immunoglobulin is increased? Because there is a lot of infection in our body, so we need antibodies to fight for them. So immunoglobulin is increased. It can also be called as hypergammaglobulinemia. So, and also increased B2 microglobulin level can be also same. So next thing I said was we have to see for infection. So there are lots of test detection for opportunistic infection we can do, and also for secondary tumors. If these three things are checked and we can find positive result in these things, then we can easily say the patient is suffering from AIDS. So that's it for the AIDS. So it is very easy topic and also very important to learn. Okay, thank you.